Welcome to the BioWhisperer channel, and the topic today is on a perspective overview of metagenomics. We will do an introduction before looking into current research applications in the area of metagenomics. Metagenomics refers to the extraction of DNA from a community so that all of the genomes of organisms in the community are pooled to study microbial communities regardless of the ability of member organisms to be cultured in the laboratory using conventional isolation. This provides an opportunity for researchers to describe the microbial diversity in environment, given that not all isolated organisms might be culturable. Sequencing the DNA of microorganisms, whether grown in culture termed as genomics or sampled from a community of microorganisms termed as metagenomics, is one of these powerful tools. In environmental metagenomics, the community of organisms is sampled from a natural environment. The earlier studies on metagenomics is done on soil samples, where two approaches are used, namely zone-based sampling and grid sampling. With each method, soil samples are collected from predefined areas in a field. Probes used for soil sampling are designed to collect a uniform amount of soil at any given depth, where proper sample labeling is also equally critical to the analyses. Microbial communities can be described in terms of levels of diversity. For instance, we can have a better understanding of the richness, the evenness and composition using taxonomic characterization of riverine bacterial microbiome. Research into aquatic sampling have shown that bacterioplankton communities vary by location and nutrient concentrations, which is a reference in the design of future studies of freshwater ecosystem health. Metagenomic approaches based on direct isolation of nucleic acids from environmental samples. Genomes are usually isolated from the environment, fragmented and then cloned into an organism by means of their plasmid, which has capacity to replicate. Organisms are then cultured to create metagenomic libraries and are then analyses based on DNA sequencing. In this workflow, apart from sequencing and genome analyses, researchers can also perform expression and functional screening. Metagenomics allows scientists to describe the genetic composition of a microbial community. These data reveal the genetic potential of organisms from an environmental sample, with a view on how microbes function and interact in natural environments. For random shotgun sequencing, where the available genomes in the sample are sequenced, we can identify novel types, to help in assembling full genomes of dominant types. This is good for communities with low diversity as well as for communities with few dominant species. From the metagenomic reads, we will form contigs, where we proceed on with the analyses. Advantage of direct sequencing is that if you use a new generation sequencing system, you can have your results in a really short time. An example of such a system is the nanopore sequencing. This revolutionary technology was first released onto the market in the year 2014. As of today, we see nanopore sequencing is rapidly becoming one of the fastest and least expensive methods for deciphering genetic variations at the molecular level. On the other hand, the disadvantage of direct sequencing meant that scientists have to obtain a really good quantity of the DNA for a representative result, and researchers may have to perform the extraction protocol many times. Metagenomic influences on disease could occur in various ways. These techniques include functional metagenomics, which serves to find new antibiotics or new antibiotic resistance genes, and descriptive metagenomics, which serves to analyze changes in the composition of the microbiota and to track the presence and abundance of already known antibiotic resistance genes in different ecosystems. Isolation of natural products from soil sampling has led to the discovery of the antibiotic turbomycin, whereas routine screening of marine microbes lead to the discovery of novel marine microbial natural products, including vitamin B, which is known to be first marine compound entering clinical trials as an anti-neoplastic agent. The second application is in the area of biosurfactant research and synthesis. Due to the increasing environmental concerns against chemical surfactants, because due to their toxicity and difficulty in being degraded in the environment, lead to the use of biosurfactants gaining popularity against their chemical counterparts. Within the context of industrial processes, such as lubrication, wetting, softening, fixing dyes, as well as in the area of making emulsions and stabilizing dispersions, which see their role in the context of bioremediation. The next application of metagenomics is in the area of biodegradation. Some of the studies carried out includes assessing the degradation of phenol and other aromatic compounds in sludge samples from a petroleum refinery wastewater treatment system or during unfortunate events, such as an oil spill. Different bioremediation techniques both upstream and downstream based on microbial metabolism, such as natural attenuation, bioaugmentation, biostimulation are promising approaches to minimize the environmental impact of oil spills. Given the presence of microbes that will naturally participate in biogeochemical roles, it is possible through metagenomic sampling, we are able to identify new genes and pathways from microbes that help us to better deal with biodegradation. Thank you for watching this video. If you like this video, please smash the like button and subscribe to this channel for future updates.